Okay, before we get to the discussion of neurotransmitters, we're going to talk about the synapse and talk about some really important work that was done by a scientist by the name of Sherrington. And it's the fundamentals of this that I think are important for understanding just how information is being transmitted from one neuron to another neuron. So again, our lecture today is about the synapse, and that's going to lead up to discussion of neurotransmitters. Here's our synapse that you see here. This is this gap between two neurons. You can see that this one is releasing some neurotransmitters out into the synapse, this gap, and it's being captured by this adjacent neuron by way of receptor sites that you see here. So we need to understand a little bit about how it came to be that we started to focus in on this gap, this synapse, which has become so important really for understanding the biology of behavior. So here is Charles Sherrington, who was a very distinguished scientist who was the first one really to coin this word of synapse, and that neurons are communicating by releasing chemicals at these junctions, at these gaps. And in 1906, Sherrington coins this term, and again, it's meant to refer to this gap that exists between neurons. And Sherrington conducts research with animals, with dogs, with cats in particular, in which he investigates basic reflexes, autonomic muscular responses to stimuli. And this work becomes really important for understanding the nature of the synapse. So let's get into a little bit about different types of neurons and take a look at what we call the reflex arc, which is, obviously very important in terms of understanding what is happening here in terms of the research of Sherrington. What you have right here is a sensory neuron. It's picking information up from the periphery, in this case the skin, transmitting that information to the spinal cord. Ultimately, it's going to go to the brain, but it's also going to connect with what are called intrinsic neurons that you see right here. Those intrinsic neurons are going to connect to other neurons, in this case to a motor neuron, to other neurons within the spinal cord. And this motor neuron that you see right here is going to be transferring information, in this case, to the muscle in the leg near the knee. So again, this is what we call the reflex arc. Okay, and it's made up of sensory neurons, intrinsic neurons, and motor neurons. So Sherrington made three really important observations that provide the basis for our understanding of the synapse and what's happening at the synapse. The first observation is that conduction along a reflex arc, which typically involves a number of different axons, sensory neurons, intrinsic neurons, and motor neurons. Typically, they're slower than conduction along a single axon. That becomes a very crucial observation. Second observation is that there are several, when we have weak stimuli presented at different points, slightly different locations or at different times, that produces a stronger reflex than a single stimulus does. And a single stimulus, in fact, may not be enough in order to provoke a particular response in a reflex arc. But when that stimulation is provided at slightly different times or on different places, it can, in fact, be enough to provoke a particular reflex arc. And third, what Sherrington shows is that when a set of muscles relaxes, another set becomes excited. So those three observations really are crucial to understanding what is happening in terms of neural transmission and ultimately what happens at this gap, this synapse. So we need to understand these important observations that he makes. So what Sherrington observes is that if you're looking at a reflex arc and you're measuring the amount of time that it takes for an action potential to travel along that reflex arc, which usually contains a number of different axons, that speed is slower than what we would see along a single axon. And he really thought that this was related to the fact that there was something that's slowing it down. And indeed, we know now that it's those gaps that slow down that whole process. So the difference then between a single axon and how rapidly that action potential occurs versus how much slower it occurs over the course of a reflex arc where there's several neurons, that difference, he believed, has to be accounted for on the basis of something that's happening at those gaps. And he believed ultimately that the evidence that he generated validated that idea of the synapse, of those gaps. So again, take a look at this very simple portrayal here. Again, we have a single axon here at the top, and we're taking a look at information traveling from here down to here, and that speed is about 40 meters per second. When you take a look at a reflex arc, on the other hand, it's slower, it's more variable. Sometimes it's in the neighborhood of about 15 meters per second or less. So again, that's much slower than what we see along a single axon. So this becomes, very important in terms of our understanding of what is happening at the synapse. So first concept that Sherrington advances is one that he calls temporal summation. And what he means by this is that if you were, for example, to take a dog and pinch the foot, the paw of the dog, and do it very lightly, doing it lightly is not going to produce the retraction of the foot. But if you do it several times in succession, then that probably will be enough to provoke the animal to retract its paw. So this is the idea of temporal summation. So you have a repeated stimulus, it's having a cumulative effect, it's producing a neural impulse. A single pinching might not be enough, but if you do several in a row, it's probably going to be enough to provoke this reflex arc. And again, it's going to be enough in order to produce this retraction that occurs in terms of the animal's leg. So here is a visual. We have a stimulus here now that's not quite up to threshold. We're going to provide another one on top of that. That still is not enough. But 3 in a row brings it up beyond that minus 50 millivolts that's needed in order to fire the neuron. So again, when we combine these in rapid succession, we get this activation that occurs, and we call this temporal summation. So now the second important proposal that he makes. He notices that when you provide small stimuli in several different locations, one stimulus might not be enough to provoke that reflex, but several at different points on the foot, pinching at 2 different points at the same time, that oftentimes is enough to cause that reflex to be produced. This is what we call spatial summation. Synaptic input from several locations accumulates and can trigger a neural impulse. So again, this is called the concept of spatial summation. So take a look at this recording, this hypothetical recording here. You're getting a stimulation from several different points. Okay, in the case of our dog, the foot, several different points, and that's going to be enough in order to get that action potential up, get that stimulation up the threshold to produce the action potential that you see here. So simultaneous pinching from different points, right? They combine spatially to get you up above threshold in order to produce the action potential. So again, this is something that we call spatial summation. Now here's a visual of temporal and spatial summation. Here you have temporal summation, you have several impulses from one neuron over time, right? And it ultimately is going to produce the action potential. In the case of spatial summation, you have impulses from several different neurons. Here's one, here's one. In and of themselves, each one is not enough, but when they're combined with one another, even though they're coming from different spots, it's going to be enough to produce this action potential. So we have what Sherrington called excitatory postsynaptic potentials, or EPSPs. They decay, they're graded potentials, they decay over time and space. But the cumulative effect of those excitatory postsynaptic potentials really provide the basis for temporal and spatial summation. So take a look at our dog now, and Sherrington, in his work, what he's going to do is he's going to be pinching, one of the paws of the dog, and what he finds is that there's retraction that occurs. But the other 3 feet simultaneously with that are going to extend, okay, here and here and here, okay. So he believed that there was the interneurons in the spinal cord were sending excitatory messages to that flexor that you see here, but simultaneous with that, inhibitory messages are being sent to the other limbs, okay, sent to the other 3 legs. So you're getting this extensor response that is occurring. So this leads to the idea then that we have inhibitory postsynaptic potentials. There's hyperpolarization that's occurring in the membrane here. And indeed, the whole concept of Sherrington that he's advancing is that these inhibitory postsynaptic potentials are really opening up these gates for these positively charged potassium ions to leave the cell, or for negatively charged chloride ions to enter the cell. So what this is going to do is this is going to produce a break, and it's going to suppress excitation, and that's why you're getting this extensor response that is occurring in the other limbs. So this really leads to the whole idea of what's called inhibitory postsynaptic potentials, or IPSPs. So when we take a look then at the characteristics of IPSPs and EPSPs, neurons can have thousands of synapses. Temporal and spatial summation can be occurring in a single neuron. When we take a look at the likelihood of any action potential occurring, it's a ratio. It's a ratio between IPSPs and EPSPs at any given moment in time. So when you take a look at that ratio then of IPSPs to EPSPs, that's going to tell you whether or not an action potential is actually going to occur. So again, these EPSPs are summating to induce an action potential. It is this equation, this summation of IPSPs and EPSPs, are ultimately they're going to cancel each other out, and they're going to determine whether or not you're going to get a firing that occurs in any given neuron. So we have what is called the spontaneous firing rate. And again, that's the production of these action potentials. And again, this is something that's occurring in the absence of any kind of synaptic input. So we have excitatory postsynaptic potentials. If you have a lot of them, many more of them than IPSPs, then an action potential is going to occur. If, on the other hand, you have a lot more IPSPs and EPSPs, it's going to cancel out the EPSPs, and the likelihood of getting action potentials, the number of action potentials below is going to be below the spontaneous firing rate. So again, it's this ratio that's incredibly important in terms of understanding action potentials. So that, in a nutshell, is the work of Sherrington, and again, how important it is for understanding what is happening with the synapse and whether or not firing is occurring. Now we need to take a look at the next step of this, which is in terms of neurotransmitters. And what happens when those action potentials get down to that presynaptic area and the release of neurotransmitters. So that will be the subject of our next lecture.